at the University of Washington. So Zack got his PhD actually at the University of Washington with Professor David Catling. And Sebastian is currently a postdoctoral researcher in David Catling's lab. So naturally, their talks are going to be topically related, which is convenient. I'm going to yield the floor now to Professor Catling, who's going to give a brief topical introduction, and then we will have a talk by Sebastian and a talk by Zack. I'm going to ask the audience that you please hold all your questions to the end. After both speakers have presented, you can raise your hand or you can type your question in the chat and we'll read it aloud. So thank you very much again, and Professor Catling, whenever you're ready.
Okay. Can you see my slides?
Yes.
Okay, good. Right. Okay, so the basic topic of interest here is environments and the origin of life. And in this brief introduction, just a few slides, because I don't want to detract from the main event. I'm just going to give a preamble as to why we got interested in things called soda lakes in particular. So in brief, I'm just going to give a little motivation as to how we came to this problem. And we came to it through something called the phosphate problem of the origin of life, which I'll describe in a minute. And earlier work that I did with a colleague published in PNAS, we identified soda lakes, which are lakes, to define them, are lakes dominated by sodium and carbonate species, as the only known natural environments currently where phosphate levels reach those that are actually used in lab synthesis of nucleotides, so the phosphorylated products of those reactions. And then what Sebastian and Zack are going to talk about is work that followed up field work to understand the controls on the water chemistry. And then Zack did some work testing the compatibility of the lake water for other things in the origin of life, in particular membranes and non-enzymatic RNA replication. And he actually did that in Jack Shostak's lab, which is at Harvard, at least it was then. Jack has since moved to Chicago. And the basic philosophy is that you can approach the origin of life with a bottom-up philosophy, which is you've got prebiotic chemistry that you can do in the lab, and you can demonstrate synthesis of particular biomolecules. But those syntheses depend on particular temperatures, pressures, pHs, solution chemistry, water activity. Maybe they involve UV light, maybe they involve particular mineral catalysts, and so on. And I'm in an Earth and Space Science department, and so I wanted to bring geoscience to bear on some of those prebiotic chemistry syntheses, in the sense of asking, well, okay, if you're using this particular water chemistry, where would you find it in a plausible environment? Because often in geochemistry, we find that one geochemical constraint sets another one. If you have this particular anion or cation, which is high, that you need for your solution chemistry, then it implies something else about the rest of the solution chemistry. And all of this bears on the when, where, and how questions for the origin of life. So one particular concern is that successful prebiotic synthesis reactions that do that make ribonucleotides and other products often use very high levels of phosphate. So a particularly well-known paper is one by Matt Pauner in Nature from 2009 that makes the pyrimidine ribonucleotides. And there the phosphate is pretty concentrated compared to natural environments. So 40 millimolars to 1 molar solution, depending on the protocol that was used. And other papers, more recent papers, have used in the tens of millimolar range of phosphate to get their reactions to work. And of course, this is fundamental because of the phosphate requirement for nucleotides, but also other important biochemicals in life, such as ATP, which you can think of as just a ribonucleotide with a few extra phosphates. And the phosphate problem of the origin of life is that I just mentioned tens of micromolar up to 1 molar concentrations, but when you normally look in natural waters, there are micromolar or submicromolar concentrations. So for example, in the ocean, typical concentration might be 0.3 micromolar. And the reason for this is that apart from biological uptake, even in the absence of biology, the phosphate is lost into an insoluble mineral, apatite, which is calcium phosphate. And on the left here, we see the see the solubility, and the axis is pretty low, even where in the more acidic conditions where it's more soluble, this is still a pretty low solubility. But in quasi-neutral conditions, essentially, it's very insoluble. And calcium is a pretty common cation, so apatite precipitates in most natural waters. And yet the high phosphate actually is not found in the more acidic waters, as you might predict from this diagram. We actually find the highest natural concentrations of phosphate in soda lakes, which are typically alkaline. And we're all familiar with this problem of phosphate, calcium phosphate solubility, because of course, with the acid that's released from bacteria in your teeth, you get tooth decay. But if you brush them and your saliva is roughly neutral, then you're okay because your teeth don't dissolve, which are made of apatite. So in this paper that we published in 2020, we did a literature survey and found that the more carbonate rich the natural water, the more phosphate rich it was, that there was a correlation here. And this data is a whole bunch of data from various lakes around the world. And the hypothesis was that the reason for this correlation is that the more carbonate you have, the more easily the calcium will be removed into insoluble calcium carbonate. And consequently, the phosphate won't be removed as the calcium, so it'll allow it to concentrate during evaporation. We'd also done some previous work showing that carbonate-rich lakes would concentrate other products, other species that are used in prebiotic chemistry, so in particular cyanide, which is often used as a starting material to get to the nucleobases. And that could come from the early atmospheres and be taken up into ferrocyanide, forms a very strongly bonded complex with iron. That can also, if the lakes evaporate, you can stockpile and concentrate that feedstock further, which can then be released into cyanide with UV or with rehydration. And so there's a nice paper summarizing that by Sasolov et al., which came out about the same time. And the idea was, well, let's simply go to these lakes that are shown up here with the highest phosphate concentration, see if this hypothesis is true in nature as it appeared to be in our lab experiments, in our models, and test this hypothesis. So the next research steps then form the basis for Sebastian's talk and Zack's talk. And this is my last slide, actually. So Sebastian's work was to test this hypothesis, go to these lakes. It turns out that these lakes here in the corner of this diagram are in British Columbia, which is handy for us because we're in the Pacific Northwest and can drive to these places. But we wanted to ask the question, what about biology? Why doesn't it remove this nutrient phosphate? After all, essentially all lakes you go to today are inhabited to some degree. And if you remove that biology, would it actually make the prebiotic version of these lakes have even higher levels of phosphate? And some of this work is published, came out just recently. This is the reference here, but there are further manuscripts in preparation. And then the second thing was to ask, well, does this lake water actually provide a suitable environment for other aspects of the origin of life? So the non-enzymatic RNA templated polymerization, lipid vesicle, membrane formation, and Zack is going to talk all about that. And this also has been published just recently, and here's the reference here. Natural soda lakes provide compatible conditions, not to take the thunder away from Zack's talk, but here it is: compatible conditions for RNA and membrane function for the origin of life. So that's really all I wanted to say, and I want to let Sebastian and Zack talk about the rest.
Thank you very much for that introduction. Sebastian, whenever you're ready.
Unmuted. All right. Let me know if there's any problems with seeing or hearing me. If not, welcome also from my side, and thank you to the organizers for putting up this really cool event on solar lakes and their potential connection to the origin of life, and for inviting us. And David, thanks for the introduction, which sets up this talk quite nicely. So I'll be talking about these high phosphate solar lakes that we've been studying for the last 2.5 years during my postdoc. And I want to share some of the insights we've gained on why these lakes are so unusually phosphate rich compared to other aquatic environments on Earth, and how the phosphorus cycle works in these lakes, and how that might inform phosphate availability at the origin of life. So I titled my talk, How Solar Lakes Could Have Satisfied Prebiotic Chemistry's Enormous Phosphate Demand, New Insights from These Modern Analogs. And before I dive right into that, I'd like to acknowledge my funding source, which is the Simons Foundation, which gave a grant to David that has financed this postdoctoral research that I'll be presenting. I'd also like to acknowledge my co-authors and all the invaluable field and lab supporters I've had who've really helped me pointing out here the awesome participants of the University of Washington Astrobiology Field Workshop in September 2023. So as David said, this research addresses the phosphorus problem of the origin of life. I'm going to be really quick because David already gave you the main points. So we need a lot of phosphate for a lot of reactions that we think are important to get life started. Perhaps most importantly, the prebiotic phosphorylation of nucleosides to make nucleotides the building blocks of RNA. And Zack will, I think, talk about how to put these nucleotides together and how he needs a lot of these nucleotides to make that work. To make a lot of these nucleotides, you probably need a lot of phosphate. Anyways, you need a lot of phosphate already just to make this reaction go forward, 10 to 100 millimolar at least, and some of the papers even more. And the problem comes in when you compare that to the environmental concentrations of phosphate, which in contemporary oceans and lakes are around 1 micromolar. So we're talking about a 5-order difference in magnitude, about 10,000 times less than we would need. So why is that? Partially it is due to biology. So that would not be so relevant at the origin of life, because biology wouldn't have been around by definition to take away phosphorus from prebiotic reactions. It is important to look to understand biology in these modern day analogs in order to then draw inferences into how phosphorus would be different in the absence of life, and it's going to be a big part of what I'll be presenting. One reason why biology takes up phosphorus, or the reason why it takes up phosphorus, is because it is still a really central element for life today, because it's needed in all these different biochemical molecules and modes like ATP, RNA, DNA, phospholipids, bones, etc. Another more directly applicable reason why phosphate is low in the environment, that's also directly applicable to the ocean of life, is what I'm saying, is the chemical side of things, where phosphate likes to form insoluble minerals with divalent cations like calcium and iron. Especially the connection between calcium and phosphate in the form of the mineral apatite will star pretty strongly in this talk. And so solar lakes have emerged in the last few years as a potential solution to this origin to this phosphorus problem of the origin of life, especially since this paper by Cohen and Catling in 2020, who, as David already said, found this connection between inorganic carbon and phosphorus in soda lakes worldwide. I want to draw your attention again to the y-axis here, which goes all the way up to into the tens of millimolar of phosphate. So that brings us, if you remember the last slide, into this area where we get enough phosphate to drive this prebiotic phosphorylation of nucleosides. So that's really important, giving us this kind of environment where we could potentially have that. And then as David said, the correlation between inorganic carbon and phosphorus tipped off Cohen and Catling to this hypothesis that apatite formation is probably low in these lakes, explaining these high phosphate concentrations because the inorganic carbon strips the water of calcium by forming insoluble calcium carbonates, and thereby allowing phosphate to stay in solution because calcium is so low, avoiding this apatite sink. And yes, so that's partially what we set out to test in the field by going to Last Chance Lake, which sits right next to another soda lake, Goodenough Lake, on the Caribou Plateau in south central British Columbia at about 1,000 meter elevation. There's an arid climate allowing these evaporative lakes to form. This gives you an idea of the scenery, of the landscape. This Last Chance Lake there in the middle, and this high plateau scenery of the Caribou Plateau. And we're looking at the landscape is made out of thin glacial till from the last ice age, overlying flood basalts. And these basalts are going to be important a few slides down the road when it comes to how solar lakes form geologically. These lakes formed after the last ice age, so can't be more than 10,000 years old, are at least 3,300 years old based on our radiocarbon dates that we got. They're closed basin lakes, which means water just flows into these lakes in dilute streams. Dilute water from streams flows into these lakes and has no other escape than evaporating. And the dilute solvents in the solutes in the stream water can just accumulate in these lakes as the water evaporates out. And these particular lakes are also very shallow. Goodenough Lake is less than a meter deep. Last Chance Lake is less than 20 to 30 centimeters deep, even in the wet season. And like I said, Last Chance Lake is the most phosphorus-rich lake that we know of, the salt phosphorus. And Goodenough Lake is also fairly phosphorus-rich, but quite a bit less. And this right there gives us this really cool natural experiment where we have these two lakes next to each other in the same hydrological setting, same overall water chemistry, the soda lake chemistry, as we will see in more detail in one of the next slides, but much more phosphorus in one of them. And so we use this natural experiment to draw conclusions as well. So generally, how to make a soda lake is pretty well understood, as what I want to show here. So you need these silica-rich minerals that occur in volcanic rocks, like these plagioclase feldspars, for example. And when these are weathered, the groundwater weathering those will have high alkalinity compared to divalent cations like calcium and magnesium. And this is pretty well understood based on a 50-year-old theory of the chemical divides that's still strong and going, and has been recently reviewed by Tosca and Tudelo and slightly updated, but it's still overall the essential theory that explains why the weathering of these silica rocks creates these high alkaline, low divalent cation solutions upon evaporation that result in these sodium carbonate and also chloride-rich brines upon evaporation. So what you need to make a soda lake is weathering of volcanic rock and evaporation in a closed basin to make an evaporative soda lake. And then we asked what makes some soda lakes extremely phosphate rich. From the beginning, we had sort of two streams two avenues that we went down to in this question, two sub-questions. One about chemistry, asking what makes phosphate so unusually soluble in soda lakes, and the hypothesis there was this lack of apatite formation, this hypothesis formed by Cohen and Catling based on this meta study. And the second question was on biology. So apparently phosphate is really soluble in these lakes, but then why doesn't biology take it up as a macronutrient, which it needs in fairly high amounts? And the hypothesis there was that biology must be limited by the second macronutrient, nitrogen. So it can only grow until it reaches the ceiling of nitrogen being used up, and then all the phosphate has to be is out of reach. Any further phosphate is out of reach. And I'm going to be talking about that toward the end of the talk, but biology will be woven into the chemistry as we go as well. And so we went to these lakes, studied them, sampled them in the early winter, in the wet season, when Last Chance Lake looks like a more or less normal lake. And then also in the dry season, when Last Chance Lake had dried up to the salt flat, where you got this thick salt crust overlying black sediments and some really thick brine and some nooks and crannies in the salt crust that we could still sample the water part of our samples from. We also sampled sediments, taking sediment cores, extracting pore water from them, did some more water sampling using weird devices to avoid contamination with stirred up sediments as we waded into the lake. And here again, a chance to thank my awesome field support. And yeah, so one of the first things we did with this with these samples is to get a general sense of the water chemistry for both lakes, and that'll also give you a good sense of what soda lake water chemistry means. Again, because these are quite typical soda lakes, they are particularly rich in carbon and sodium, and also quite rich in chloride. They are moderate. Potassium, magnesium, and sulfate are moderately abundant, and they are fairly poor in nitrogen, calcium, and iron. And the big difference between the two is, as I said, the much higher phosphate in Last Chance Lake, and also Last Chance Lake is quite a bit more saline, up to 13 times seawater salinity versus 1 to 2 times seawater salinity in Goodenough Lake. But that salinity is not simply the reason for the higher phosphate in Last Chance Lake. So if you multiply up the difference in salinity, apply that to the phosphate, multiply the difference from salinity with the Goodenough Lake phosphate, you get nowhere near to the phosphate that we find in Last Chance Lake. It's much lower. And this is what phosphate looks like across the season. So these are all samples from the different seasons. And on the x-axis, we see chloride, which stands in for salinity. It's a conservative salinity tracer. And so the more as we evaporate the lake from the wet season to the dry season, from left to right, going higher in salinity, phosphate just also increases. There's this direct correlation, but a pretty strong correlation with salinity, indicating phosphate in Last Chance Lake behaves like a conservative salt, conservative iron. And in the dry season, we find these really high phosphate concentrations in this really thick brine that we sample in the salt crust. In Goodenough Lake, we don't have this conservative behavior of phosphate. Phosphate gets pretty high if you compare it to your typical freshwater lake. There's a couple of samples around 1 millimolar, but it doesn't correlate with salinity, meaning there's something else going on. And that is probably biology, or I can give away it's biology, and we look into how exactly biology does that in Goodenough versus Last Chance Lake later on. For now, let's look at the mineralogy. So we looked at the water chemistry before. Now we look at the solids. Kimberly Sinclair, who is a PhD student in David Catling's lab, she looked at the minerals in the salt crust and sediments using X-ray diffraction. And in the salt crust, she found sodium bicarbonates, mostly like thermonatrite, trona, berkite, and nacolite. And in the sediment, she found detrital minerals, so these feldspars that are necessary to be weathered in order to create soda lakes. So that makes a lot of sense that they are around. And she found dolomite, which is a calcium magnesium carbonate, and that tells us where the calcium goes. So remember, the water column is stripped of calcium, and it is indeed stripped of calcium by inorganic carbon, forming calcium carbonates in the form of dolomite specifically here. And so this dolomite finding in the sediment makes a lot of sense. It's fairly insoluble compared to these sodium carbonates. and precipitates out early in the evaporation sequence. So as the lake evaporates, dolomite is one of the first to go. So it ends up in the sediments rather than the salt crust. For phosphorus, we can't say much using XRD. The detection limit is too high, so phosphorus is too low to be detected by XRD, and we have to use a chemical approach to get an idea of the types of phosphorus fractions we find in the sediments of the lakes. For that, we take a sediment sample and apply this method by Rottenberg 1992, where we throw stronger and stronger solvents at the sediment sample and extract different fractions of phosphorus, including loosely sorbed phosphorus, iron-bound phosphorus, orthogenic apatite, tridal apatite, and organic-bound phosphorus. And of these different fractions, I pointed out the three that could be phosphorus sinks, meaning they are fractions that would be formally dissolved phosphorus that formed one way or another a precipitate, for example, a mineral precipitate like orthogenic apatite, or a biological precipitate by binding or being taken up by biology as organic bound phosphorus, and then end up in the sediments, or they could form in the sediments directly and get buried there and removed from the system, becoming a phosphorus sink. So phosphorus removed from the system. And we were interested in that, and we were able to quantify that also using sedimentation rate estimates to quantify these phosphorus sinks in the context of a phosphorus budget, where we then also quantified the sources of phosphorus to the lake, mainly the phosphorus in spring water inflow, so these streams and springs flowing into the lake, where we measured the phosphorus as well, and we inferred the water inflow rate from meteorological data, as well as things like evaporation and precipitation. Atmospheric phosphorus deposition is a minor influx of phosphorus to the lake. So this budget compares the sources of phosphorus to the sinks, and then comes up, what it comes up with is an excess of the sources over the sinks, so a net phosphorus influx into the lake on a time basis, like a year, so a net annual phosphorus influx. And this is a very complicated figure. You don't have to understand all these numbers. They will be eventually published, hopefully. And I'll make it slightly more simple and point out what I really want to get at here is the sink for apatite in these lakes is really small. It's the smallest of the three sinks that we looked at. And it's also small if generally the phosphorus sinks small compared to other comparable environments, maybe like the ocean or freshwater lakes. There's very little phosphorus export, and the orthogenic apatite sink is particularly low. And that, of course, is consistent with this initial hypothesis by Cohen and Catling that low apatite formation is a reason for the high phosphate concentrations in solar lakes. Organic phosphorus sink is also really low in Last Chance Lake. And next slide, we look at the same thing for the same budget for Goodenough Lake, finding much more organic phosphorus export. Also, fairly low orthogenic apatite sink. It's also the smallest of the three sinks in Goodenough Lake, slightly higher than in Last Chance Lake. But we can conclude from this, so 20 times more organic phosphorus export, we can conclude from this that the lack of apatite, the apatite sink is in fact low in both lakes, allowing for this high millimolar phosphorus solubility in both lakes. But the organic phosphorus burial is what distinguishes the two lakes, because it's much higher, I should say, much higher here, not much lower. So the organic phosphorus burial is much higher in Goodenough Lake, distinguishing the two lakes, whereas both have this low apatite sink. And so getting a bit further into the role of this organic phosphorus burial, what I did here was to plot this annual phosphorus budget. So the annual the net phosphorus influx into Last Chance Lake and Goodenough Lake. Plot that as a function of time. So on the x-axis we have lake age between 1,000 and 10,000 years. So you can just sum up the annual net influx of phosphorus and come up with this timeline. Okay, and the relevant time frame here is between 3,300 and 10,000 years, which is the age of the lakes that that we've narrowed down their lifetime to. And so you see that after in this time period, Larsen's Lake has accumulated these 4 millimolar of phosphate. This is for the wet season model for the wet season water column, and that's what we find in the water column in the wet season, around 4 millimolar. And so then what I can do in this model is to switch off the organic phosphorus sink. And if I do that for Larsen's Lake, even though the organic phosphorus sink is small, we still get, of course, over time, a little more phosphorus accumulation because we switch off this small sink. In Goodenough Lake, where the organic phosphorus sink is high, we start at this very small baseline because the phosphorus budget in Goodenough Lake is very small, accounting for its much lower dissolved phosphate concentrations. But if we switch off the organic phosphorus, we find a lot more phosphorus because of the large organic phosphorus sink that we're switching off, and we get phosphorus accumulations over several thousand years at the order of magnitude of Larsen's Lake. And that suggests that without biology, Goodenough Lake and probably many more solar lakes might have millimolar level phosphate concentrations. And it could also implicate that the extreme salinity that some people are bothered by in terms of the extreme salinity of Larsen's Lake, up to 13 times seawater salinity, could be a problem for some chemical reactions that could be important for the origin of life. Maybe that's actually unnecessary because Goodenough Lake only has 1 to 2 times seawater salinity, and perhaps without biology we would have similar phosphate concentrations in around 10 millimolar without the biology. And so that finally brings us to the reason to how biology actually does that. Why is it so active in Goodenough Lake, but not in Larsen's Lake, apparently? Why is there all this organic phosphorus in Goodenough Lake and not in neighboring Larsen's Lake? And why doesn't biology in Larsen's Lake take up phosphate as much as it seems to be doing in Goodenough Lake, in other words? And we had this hypothesis of the limitation by nitrogen, and we tested that quite directly with this incubation experiment where we took water from each lake and added a nitrate ammonium mixture to it. And over 48 hours, we got, so we incubated that in sunlight, natural sunlight. And over 48 hours, we got chlorophyll A to increase quite significantly in Larsen's Lake following nitrate ammonium treatment, but not in the controls where we treated with phosphate, or control which we didn't treat with anything, and also in Goodenough Lake, no response, indicating quite clearly that Larsen's Lake phototrophic biology is nitrogen limited, but we didn't find the same evidence for that in Goodenough Lake. And then the next question is, so why? Why isn't Goodenough Lake's biology also nitrogen limited? So we measured nitrogen fixation rates, which is the nitrogen fixation being this process of this microbial mediated process where these specialized microbes can take the abundant N2 from the atmosphere, 80% of the atmosphere is N2, and turn it into biomass. It's a specialized high energy process, for example, done by many cyanobacteria. And there are in fact cyanobacterial mats in Goodenough Lake. And we sampled those for these incubation experiments, as well as sediment and water from Larsen's Lake, and found much higher nitrogen fixation rates in these Goodenough microbial mats compared to Larsen's Lake, where there's no such mats. So we're comparing that to sediments in Larsen's Lake, and we also looked at the water column and finding much higher very high nitrogen fixation rates in these microbial mats in Goodenough Lake. That can actually explain this phosphate demand by biology. So here we removed the nitrogen ceiling, or we burst through the nitrogen ceiling using nitrogen fixation and being able to make use of that phosphate that would have otherwise accumulated probably in Goodenough Lake as it does in Larsen's Lake to really high concentrations. All right. One more question that we haven't quite solved, but have a good idea of, is why this different in nitrogen fixation now? So why much less nitrogen fixation in Larsen's Lake than in Goodenough Lake? And here, the best hypothesis we have is the high salinity, and we base that hypothesis on again, metadata from solar lakes worldwide. There's not a whole lot of nitrogen fixation rates having been measured in solar lakes worldwide, but the ones that have been measured, the high rates that we need in good enough lake to balance these high phosphate concentrations in good enough lake, these high rates we don't find above a certain salinity that is applicable to large chance lakes salinity. So the high salinity may repress the nitrogenase enzyme synthesis, so the enzyme facilitating nitrogen fixation, and could also divert energy away from this energy-intensive process of nitrogen fixation. That remains to be finally tested, though. And so finally, so what's needed? So starting to summarize this whole thing, what's needed to make a high phosphorus solar lake these days, and that these days refers to biological Earth. So we need limited biological uptake, like in large chance lake, where it might be salinity inhibited. We need this geochemical solubility due to in this case, in the case of solar lakes, due to the lack of calcium that's been stripped out of the water column by carbon, and this low orthogenic apatite sink resulting from that, allowing phosphate to stay in solution. We need some, to some extent, we need evapoconcentration. Some of our results there might indicate that we don't quite need this extreme evaporate concentration in large chance lake to get really high phosphate concentrations in around 10 millimolar. But even good enough lake has 1 to 2 times seawater salinity. So some evaporate concentration is definitely the case in both lakes. And then finally, we need time. So these lakes have evolved over several thousand years. And one thing that I haven't maybe emphasized as much yet is that you can't just, so if you look at the phosphorus budget of the lakes, let's take large chance lake, and we look at the amount of phosphorus flowing into the lake per year, that's much less on a square meter basis or liter basis than we find phosphate dissolved in large chance lake. You need to get to those levels, you need centuries, probably millennia. And then there's also an interesting side to that. So it could be possible that lakes, that older solar lakes than large chance lake, or solar lakes could become older than large chance lake has become so far, and could potentially even accumulate more phosphorus than we see in large chance lake today. And so just to throw up my main points that I've made in this talk for our discussion later on, perhaps. So we confirmed this lack of an apatite sink, maybe that's a bit strong, so a very low apatite sink in solar lakes. That is the basis for phosphate solubility in solar lakes. We showed how and that biology suppresses phosphorus concentrations in good enough lake by taking them up and making its way to it by fixing its own nitrogen. So inhibited biology is key to forming extremely phosphorus lakes on modern Earth, which also implicates that the share of high phosphorus solar lakes was likely higher on prebiotic versus modern Earth. So out of all solar lakes on modern Earth, only a few are really high phosphorus, let's say millimolar, and on a prebiotic Earth without biology, this share may have been much higher than what we see nowadays. And then there's indications that we might need less saline conditions than the extremely saline conditions in large chance lake to get this phosphate. Generally, large chance lake is a surprisingly good, if not perfect, prebiotic analog because of its low biological influence, potentially inhibited by salinity, and that gives us a pretty surprisingly good analog on biological Earth, where chemistry is dominant in large chance lake, despite the biology that tends to be in most places on Earth, and also in large chance lake, just not as influential. And that gives us a pretty good window into maybe a prebiotic solar lake. And finally, I'm currently at the University of Cambridge working in Nick Tosca's lab here with Clancy Chang on some things that we can't directly see in large chance lake when it comes to prebiotic chemistry. The big problem with large chance lake when it comes to prebiotic chemistry is that it has an oxic water column. And so some things that are redox sensitive, like iron, are definitely not very analogous in Last Chance Lake. And so here we're looking at the role of iron that might be overlooked in just looking at Last Chance Lake today, where iron would, in the water column anyways, would be in the oxidized form, iron 3. And here we're looking into lab experiments with reduced iron under anoxic conditions and how that might alter, although we suspect that it probably doesn't significantly alter the story, but that's something we're testing here, how it might alter the story of phosphate solubility in soda lakes. And with that, thank you and happy to pass on to Zack.
Sebastian, that was very interesting. And now Zack will continue the story and tell us how conditions like these may have facilitated the development of early life. Take it away, Zack.
Okay, let me share my screen. Just gonna try and move some zoom bars around. Okay, yeah, I'm Zack Cohen. I'm currently a postdoctoral fellow at NASA Ames Research Center. And the project I'll tell you about today, of course, happened when I was a graduate student at University of Washington, working with Sebastian and with David Catling and many others. So the title of the talk is Natural Soda Lakes Provide Compatible Conditions for RNA and Membrane Function that Could Have Enabled the Origin of Life on Earth. So a bit of a mouthful, and I'll try to walk us through it. So the goal of this research has been to identify plausible environments for the assembly of cells on the early Earth. So of course, I'm going to be telling you guys about soda lakes, and specifically 3 stories about these lakes. First is about non-enzymatic synthesis of RNA by primer extension in natural lake water. Second story will be about the assembly of RNA by ligase ribozyme in the lake water. And then the third story will be about encapsulation by fatty acid vesicles. So to start with some background, I want to ask, why has it been so challenging to identify natural environments where the first cells could have assembled? And I think there's 2 reasons for this. The first reason is that we are uncertain about sources of building blocks in these natural environments, building blocks like nucleotides and fatty acids. And what I want to tell you guys about this is that I think soda lakes are promising places to have, we have some ideas about how they would have concentrated all of these different cellular components in the same single spot. So first, thinking about nucleotides, the synthesis of nucleotides, we're sort of guided by this pathway from a few years ago from the Sutherland lab, where they have shown that through this sort of series of reactions involving phosphate, ferrocyanide, and light, you could have generated nucleotides abiotically. Of course, some key things here, right? Of phosphate, like Sebastian has told us about, ferrocyanide, and light. These are things we think could have actually been available in these soda lake environments and are important precursors to nucleotides, potentially. Now, the second thing I'll tell you about is membrane formation. So we know that one source of amphiphiles of fatty acids on the early Earth is delivery by meteorites. And if we imagine that, one of these meteorites happened to land in a soda lake on the early Earth, what we can estimate based on modern meteorites landing to Earth is that those fatty acids in the meteorite would have been too dilute to assemble directly into membranes. So if we imagine that membranes formed from fatty acids delivered by meteorites, we need some concentrating mechanism like evaporation that is provided by these lakes. So after evaporation in the dry season could raise the concentration of those fatty acids above the critical vesicle concentration, and we imagine that we could have membranes. Similarly, with forming oligomers of nucleotides, so RNA oligomers or peptides, it's been shown in lab experiments that cycles of wetting and drying can be very useful for building these oligomers. And this is something that these lakes experience, right? With their dilute wet seasons and then transition into the dry season and cycling through years. So I would say, yes, especially compared to other environments we might imagine for the early Earth, that we have at least ideas about plausible sources of building blocks in these soda lakes. The second problem facing the assembly of cells is the distinct requirements for some of these components, such as RNA and fatty acid membranes. And really, I'm talking about divalent cations like magnesium. We know that divalent cations are important for RNA structure and for many reactions involving RNA, but even adding like a little bit of magnesium, 10 millimolar magnesium to fatty acid vesicles can completely destroy them. Whereas on the left, we have this picture of some very nice spherical vesicles without any magnesium. So from some lab experiments, we have an idea about how to have compatible conditions for RNA and for fatty acid membranes. So the first way to achieve this has been shown in a number of experiments from the Shostak lab, is to use a relatively high concentration of citrate, sodium citrate, to chelate a relatively high concentration of magnesium, 50 millimolar magnesium. And what they have shown is that this will allow the magnesium to still sort of interact with the RNA and stabilize RNA, but it stabilizes the membrane too. It prevents destruction of fatty acid vesicles by the magnesium. A sort of problem with this idea is it's not obvious what the natural source of such a high concentration of citrate would have been. So the second set of conditions that people have used is just to keep the magnesium concentration relatively low, less than 4 millimolar. And this is nice because this is basically what soda lakes really have in their natural conditions. So as we discussed, Sebastian discussed during his talk, some of the most abundant anions in this lake water are carbonate and bicarbonate. And because the solubility of magnesium carbonate and other carbonate salts of divalent cations, because that solubility is poor, these lakes have an upper limit of about 1 millimolar magnesium and 1 millimolar divalent cations. So my hypothesis then is that soda lakes have enough magnesium for RNA reactions, but not too much magnesium to destroy fatty acid vesicles. So in order to test this, right, I traveled to these lakes with Sebastian and David and others from University of Washington. And we collected samples. We collected samples during multiple seasons. The samples that I'll focus on are from the dry season, when this lake water is saturated with salts. So the rough composition is about 1 molar of monovalent cations like sodium, 1 millimolar divalent cations like magnesium at pH 10. So then we brought that water back to the lab, and we wanted to test these potentially important steps for the assembly of the first cells using this water from the natural lakes. So the first story I'll tell you guys about is non-enzymatic synthesis of RNA by primer extension. And the reason we're interested in this is, if we imagine early cells based on genomes of RNA, we can ask how could these RNA genomes have been replicated before the emergence of enzymes? So the first thing we might imagine is that once we have nucleotides forming, that we can form these random short oligomers of nucleotides, these random sequence RNAs in the environment. And then if there's any complementary sequence between these RNAs, of course, we'd expect base pairing. And we call the short piece of RNA the primer, and the long piece on the bottom the template. Then what we imagine is that free nucleotides from the environment can come in, base pair with the template, react with the primer to extend the primer by 1 nucleotide. And then this could happen again, right? The next nucleotide comes in, base pairs with the template, and extends the primer until we have full extension of the primer on the template. then imagine some mechanism to separate the strands. And so now, effectively, what's happened is we have synthesized the complementary strand of our original RNA. Maybe that RNA, does something interesting, and so we want to make copies of it. So then you could imagine going through this whole process again on the complementary strand to make a copy of the original interesting RNA. And so the part of this that I'll focus on with my experiments is this primer extension part of the process. So will RNA primer extension work in natural water from these soda lakes? In order to test this, I traveled to Boston to work with some really amazing collaborators in the Shostak lab. And this is the procedure that we came up with to test it. So the first thing we did was to filter the natural lake water so that no cells, no sediments remain, just all the dissolved ions. Then we added RNA primer and templates. And then in step 3, we add a solution of activated nucleotides to initiate the reaction. And here we were using nucleotides activated with 2-amino-imidazole instead of triphosphates, which are more common in biology. I'll talk briefly about that in a little bit more detail. But what you need to know in step 3 is we initiate the reaction by adding these nucleotides. And then in step 4, we'll take aliquots of the reactions and quench them and run them on a gel. So here is the first finding, that the rate of adding the first nucleotide onto the primer is comparable in lake water as it is to standard lab conditions. This was surprising and I think exciting. So I'll show you the data. On the y-axis here are the pseudo first order rate constants for this reaction for extension by the first nucleotide. And in lab conditions, which are 50 millimolar magnesium at pH 8, this is what we measure here. And then in our natural waters from Last Chance Lake and Good Enough Lake, we measure a little bit lower, but really comparable, not orders of magnitude lower like maybe you would have expected naively. And I also prepared an artificial lake sample that has half molar sodium carbonate and 1 millimolar magnesium chloride at pH 10. So roughly simulating the salt composition of these lakes. And that sample had the highest initial rate of reaction. So again, this was encouraging, right, for our idea that these kinds of reactions might have worked in natural lakes on the early Earth. So after 3 days, now the story is a little bit different. So now we're looking at the percentage of all of our RNA primers that have been extended by 3 nucleotides. So that's what's on the y-axis here. And in lab conditions, that's nearly 100%. But in our lake samples, it's much lower. So this was a bit disappointing, right? This is saying even if the reaction's fast initially, it seems to slow down quickly for some reason. And we would imagine that's, wouldn't have been good for early cells developing in these lakes. But what we found is that the yield of these extended primers can be increased by adding a larger initial supply of nucleotides. So the y-axis here is the same as the last slide, the percentage of all our primers that have been extended by 3 nucleotides. And in lab conditions, we have nearly 100% extension regardless of the initial nucleotide concentration. But for our lake samples, what we now see is if we use 100 millimolar nucleotide instead of 20, the yield of the reaction doubles in some cases. It's almost double in some and increases substantially. And so this is encouraging. I mean, this would say that maybe on a per nucleotide basis, the reaction is less efficient in lake water than it is in some other conditions, but we can reach these high yields as long as we keep the nucleotide concentration high. So I'll make one more comment about these sets of experiments, a technical comment, I guess, for people that are familiar with this. So here I'm showing one of these activated nucleotide molecules. So we have the nucleotide in black, and then this 2-amino-imidazole molecule activating on the phosphate. And what's been shown in laboratory conditions is that two copies of this molecule can react together to form this bridged dinucleotide intermediate. And that really it's this bridged dinucleotide that comes in and pairs with the template and reacts with the primer to extend the primer by one nucleotide. And what I'll tell you here is that basically this is the mechanism that's been figured out in lab conditions. And it seems like the same mechanism is working in the lake conditions as well. So that's the first story about non-enzymatic synthesis of RNA by primer extension. The next thing we were interested in was, can RNA be assembled by ribozymes? So here's a schematic of the reaction that we tested, where we have a ligase ribozyme paired with a template RNA in gray. And this template RNA is also paired to a substrate RNA. And the substrate RNA is activated with 2-amino-imidazole as well at the 5' end. This molecule that we know works well for the reactions and maybe was more available than triphosphates on the early Earth. So anyway, I set this reaction up in 75% water from the natural lakes, and after 20 hours did not observe any reacted product. So that was a bit disappointing, and we thought about why is this happening? And we realized that this ribozyme was selected to work at pH 8, and our natural water is at pH 10. So the next thing that we tried is to bubble in carbon dioxide into our natural lake water, bring the pH down from 10 to 9, and then set up the reaction again. And after 20 hours, now we do actually observe ligation. So that I think that's encouraging, but maybe you'll say to me, so what? You modified the natural lake water. This is not that exciting. And I would say two things to that. First, our modification to decrease the pH might actually be more in line with what we expect of soda lakes on the early Earth because of a high partial pressure of carbon dioxide in the atmosphere, which might suppress the pH. But the second thing that I would say is, I think that if we selected ribozymes in this natural lake water, or if we imagine natural selection on lakes on the early Earth, then I think you could identify ribozymes that would function optimally in the unmodified lake water. I think so this experiment is kind of a proof of principle that it can work. So the third story will be about encapsulation by vesicles. So here on the left, I'm showing decanoic acid, which is a fatty acid that has been delivered to Earth by meteorites. And we imagine there would be other abiotic sources for this fatty acid as well on the early Earth. And we know that dissolved in water at the right pH, we have a mixture of protonated and deprotonated decanoic acid molecules. We know that these can assemble into vesicles that will have a net negative charge on the surface. So if I add a lot of sodium cations, effectively that would screen the charges on the surface and cause these vesicles to aggregate with each other due to the hydrophobic effect. If they're uncharged, there's no repulsion between the vesicles. So that's the effect of monovalent cations like sodium. If you have a lot of divalent cations like magnesium, then those can chelate adjacent fatty acids and bring them out of solution, and it's much more disruptive to the vesicles. So in past experiments where I used artificial sodium solutions, I saw that vesicles still retain their encapsulated contents in artificial solutions with a lot of sodium. So here, walk through these images. I prepared vesicles with an aqueous fluorescent dye encapsulated and a hydrophobic fluorescent dye in the membrane, a different dye in the membrane. So then I'll add enough salts, sodium chloride, I think in this case, to completely saturate the solution with sodium. And then now what we see are these big clumps. So if you look at the scale bar here, this is a zoomed out view, and we have these big clumps, which are many, many vesicles aggregated together. But what's interesting in these images is that despite this clumps, right, this process of salt-induced flocculation, these clumps are still bright with the aqueous dye, basically suggesting that the individual vesicles are retaining the dye. And when you dilute the salts, individual vesicles separate from each other, and many of those vesicles still retain the dye. So despite salt-induced flocculation in artificial sodium solutions, vesicles maintain their encapsulation. And basically, we saw the same exact thing in natural lake water, despite now having a little bit of divalent cations like magnesium. So here, again, I prepared vesicles with a fluorescent dye inside, and then replaced the exterior solution with water from Last Chance Lake. And because this water has a lot of sodium in it, the vesicles aggregate into the big clumps, right? Salt-induced flocculation. And still those clumps are bright with the aqueous dye. Dilute the salt, individual vesicles separate from each other. Many of those vesicles still retain the dye as well. So this is encouraging for imagining protocells developing in these lakes. And I want to now go a little bit deeper, tell you guys a little bit about the details about how I did these experiments, because I think it's informative. So the first thing that I did was to add my amphiphiles, fatty acids and fatty alcohol, into a dilute salt solution with fluorescent dye on the inside and the outside. Then in step 2, I add 2 volume equivalents of natural lake water. And because of all the sodium, right, the vesicles clump together, float to the top, and then I can remove most of the solution below that vesicle layer and go back to step 2, add 2 more volume equivalents, float the vesicles, remove the solution, the lower solution, and repeat that whole procedure 5 times until basically there's no dye left in the exterior solution. And this tells me, okay, I've done a pretty good job of replacing that exterior solution with natural lake water. So that's how I prepared the samples that I just showed you. And here's actually what the test tubes look like. So initially, we have this sort of uniformly cloudy solution because the vesicles are dispersed in this dilute salt solution, dye on inside, dye on the outside. But at the very end of this replacement process, now I have this layer of flocculated vesicles at the top here that is very bright with the dye compared to the exterior solution, which has little dye. So that's all what happens when I assemble the vesicles initially in dilute salt solution. Instead, if I try to add the amphiphiles directly to water from the Last Chance Lake dry season, then the tube looks similar initially, but after going through this whole replacement process of the exterior solution, now what I see is a very thin layer at the top of my sample that I think is just sort of aggregated amphiphiles. They're not assembled into vesicles, and they don't have any encapsulated dye. So the amphiphiles really need to assemble into vesicles in dilute solution, and then they can tolerate the salty periods, but they can't assemble in salty solution. So this led us to think about this sort of life cycle for protocells developing in these lakes, where we imagine that new vesicles could form from free fatty acids during the wet season. And as has been seen in lab experiments, those vesicles can grow and divide. But during the dry season, flocculation doesn't disrupt their encapsulation ability. So they can the vesicles can, survive these transient dry periods and wait till the next wet season to do their growth. But in the dry season is where we have seen that we can have non-enzymatic RNA copying, or at least primer extension, and ribozyme activity as well. So that's 3 stories about these lakes that I think, the upshot, I think, is that these natural soda lakes are promising sites to imagine the origin of life on Earth. So with that, I want to thank all of my collaborators from Mass General in Boston and from University of Washington. This was a very collaborative project, and I really relied on the expertise of all of these people. So I really want to acknowledge them. And thank you guys for listening. Thanks to the organizers for inviting me to talk. And I'd be happy to take any questions.
That was very interesting. So now we'll have the Q&A session for both speakers. What you can do is raise your hand in the Zoom interface if you want to say your question aloud, or you can type your question into the chat and we'll read it aloud for you. Okay, so Kevin has a question, so go ahead.
Yeah, thank you, Sebastian and Zack, for very interesting presentations. My question's very more towards Zack. What was the max on your sort of like non-enzymatic sort of like RNA extension? What was the maximum length of primers that you were able to make? Secondly, did you try varying the pH of the water of the sort of lab water? Did you go down to say 7.5 or 6? And if so, what difference did that make? And finally, are you interested in actually using natural triphosphates rather than amino imidazole phosphates as building blocks, maybe in the presence of some borate minerals or another inorganic catalyst?
Yeah, thank you for asking that. Those are all great questions. About the max length. So we had a primer, and the template had an overhang of 4 unpaired nucleotide positions.
Mm-hmm.
And so I was telling you about mostly extension by 3.
Yeah.
The extension by 4, this is the problem called the last base addition problem that has been observed in laboratory conditions, basically because of the formation of this bridge dinucleotide that I talked about. It doesn't have the ability to pair with at two positions on that when there's only the final position on the template. So that's been known to be less efficient. It's also less efficient in the lab conditions. How much of that form? I think it's. Let me see if I have a slide on that. It's a much lower percentage that actually gets fully extended by +4. Yeah, it's yeah, I think it's only like 5% that gets extended by +4 in natural lakes. Let's see. Second question was about
the pH. Varying the pH.
Yes. Okay. So we did artificial solutions and varied the pH and observed that yes, that I think that accounts for a lot of the different the fast rate that we see in the soda lake waters, that the pH, the reaction is a lot faster at high pH. Yes. And your third question was about
amino imidazole phosphates with natural triphosphates to see if you do get any non-enzymatic primary extension and ligation.
Yeah, I think that it's been tested pretty well in lab conditions, and that the reaction is quite slow with the triphosphates. But it would be interesting to test it in the natural lake water. I mean, like these are very different conditions, and if it was a lot faster than lab conditions in natural lake water, that would be exciting. But that's not something I have tested. But I agree it would be interesting to test.
Okay, thank you. Thanks very much. Thank you.
Thank you.
Next question is from Eduardo. You can go ahead and unmute yourself.
Hi. Yeah, I was wondering, had you guys considered the atmospheric content of oxygen, how that would change the chemistry of the crust over 2 billion years, considering that prior to life, the minimal carbon dioxide content of the atmosphere was at least 10%. Wouldn't that 2 billion years of oxygen content on the crust change the type of minerals that are going to be generated and the chemistry that's going to occur between the oxygen? The oxygen and the lakes. Whereas prior to life, there was such an extreme content of carbon dioxide, and that the minerals generated during that time with the absence of oxygen would have been different, and those lakes would have had different chemistries.
Sebastian, do you want
to take this? Yeah. Okay. So So I think it would probably actually work in the direction of making it more phosphorus. So the things I would think of about having more CO2 in the atmosphere in prebiotic times would first of all the lakes would more easily become inorganic carbon rich. And then weathering rates might be higher. There's been papers, I think, by Howe and colleagues showing that weathering rates and would have been higher on early Earth due to these more acidic conditions in the high CO2 atmosphere. That could have led to higher phosphate concentrations, sort of on average, in like streams on prebiotic on early Earth compared to today. So that could actually all sort of work toward having even
Benefit.
likely higher phosphate concentrations in an early Earth version.
Awesome. Because I was worried that, we're doing experiments with phosphorus lakes now, and we are seeing some results, but I like you, I would think it would be a much more uh, conducive environment with more carbon dioxide in the atmosphere. Okay, thank you.
Yeah, and I guess I would say about the lab experiments, it's not obvious to me how the presence of oxygen would change things, but I mean, obviously it's harder to do those experiments in an anoxic environment, but I think it would be interesting. Yeah, I mean, I wouldn't be that shocked if it did change things. So I agree that.
The presence of oxygen would alter the formation of the minerals, especially from a molten state, and the extrusion and the induction of the crust being done in the presence of oxygen would create different types of minerals.
Right.
But again, like you said, with the increase in carbon dioxide, that only aids the formation of said phosphorus, uh, dilution of phosphorus in. Thank you. I just wanted to know. Thank you.
Yeah, I have something to add actually to this. So one thing that provides a bit of uncertainty in this regard is the sulfate that's in these soda lakes in Large Chance Lake and Goodenough Lake. That is probably unrealistic for prebiotic soda lakes because it probably comes from metamorphic rock weathering. There seems to be a little bit of metamorphic rock influence in these lakes that wouldn't have been, so it would have been all volcanic rock, basaltic rock on early Earth. And so that sulfate could actually work toward I don't actually think so at the end of the day for solar lakes, but so the role of sulfur in all this would be that sulfur in the present solar lakes binds the iron as iron pretty
Right.
insoluble iron sulfides, and that's something that's unrealistic for early Earth. But at the end of the day, it probably doesn't matter. So it's partly like what I'm looking into now in these lab experiments here, because the iron should probably be taken care of by the carbonate as well. So forming iron carbonates is our hypothesis.
Right.
But yeah, maybe something to consider as well, the sulfur. That's
all the iron that we have in all the bridges, all the cars and all the buildings we've constructed, all that iron was diluted in the ocean prior to the oxygenation. As evidence, I mean, the iron we get is from the iron band that occurred once oxygenation started on Earth, and all that iron was precipitated out of the water and formed that layer that we use now for modern day iron. So, and that again, all that iron diluted into the water benefit as it does benefit photosynthesis. So again, it would have been much better. Prebiotic chemistry would have been much more conducive to life prior to the oxygenation of the atmosphere. Well, I'm done. You can mute me. Thank you, gentlemen.
Thank you.
Thanks.
So I have a question for both speakers. So as probably everyone in this audience knows, the incorporation of phosphate into early life is one of the biggest problems in understanding the origin of life. And it's a multifaceted problem. I think the two main components of it are the mobilization of phosphate, as you spoke on, and the activation of phosphate in order to undergo substitution by nucleophiles. Now, there are a lot of proposals in the literature about how that might have occurred, and a lot of them are mutually exclusive. Some of them are under acidic conditions. Some of them they invoke high concentrations of urea or formamide. Others invoke environmental generation of condensing agents and so on. Does your research tell you anything about that problem? Do you have an expert opinion on how the initial incorporation or activation of phosphate may have occurred?
I don't think I have very good ideas about that. I mentioned, I guess, the pathway from the Sutherland group to make nucleotides. I know that they're thinking about how to imagine some of those steps in these kinds of environments, but I don't think I have a great answer to that question. It's a really good question.
No problem. It's interesting because the lake mobilizes phosphate to such a high degree, and that seems to give you a foot in the door into incorporating the phosphate. Sebastian, do you have any thoughts on it?
Yeah, also basically, I'm influenced also by the Sutherland group ideas, and would add, and I had a slide in there at some point that I threw out, that they have this scenario that's quite compatible with sort of a Salt Lake setting in the Patel et al. 2015, I believe, where they imagine these little streams coming together, which have different conditions and may have formed some intermediary or another that meets the other one and then eventually flows into this lake. And that's the kind of setting that invokes a soda lake, like little streams flowing into a soda lake is quite compatible with that. So yeah, it's also very attractive to me, that kind of scenario. Yeah.
The paper that has the word cyanosulfidic in its title, right?
Yes.
I would just add that, apart from high levels of phosphate to drive by Le Chatelier's principle reactions to the right, in Matt Pauner's chemistry, phosphate is also an acid-base catalyst. And if I remember rightly, what it's doing is it's catalyzing hydrolysis of a product in that scheme to make urea. And then the urea itself promotes phosphorylation. So, there's a catalytic role, and there's also just the fact that you've got a lot of phosphate. So I think there's several ways to think about this, but I'll leave it to those guys to defend their prebiotic organic chemistry. But what I'm trying to do is say, okay, that's what you use. How do we find that in a natural condition? I also have to leave right now because I've got to go to another engagement. So sorry about that.
Sure, so thank you. So Kevin has one more question. Go ahead.
About I presume I also put it in the chat box about the prebiotic synthesis of ribonucleotide triphosphates and polyribonucleotides on prebiotic glasses by Stephen Benner's group. I presume you guys are quite familiar with that. And those experiments were carried out though at pH 7.5. That gave the best synthesis of polyribonucleic acid, and they know it's definitely RNA from classical analytical experiments done using cleavage with sodium periodate, which was actually used. That was the actual experimental technique used back in the 1950s to identify the actual structure of RNA nucleosides. Are you familiar with that? I presume you're familiar with those papers from 2 years ago.
Yeah, very interesting paper.
That's it. I mean, that would favor, I would think, though, if that's the case, that would be like early Earth conditions, but it would favor more neutral aqueous environments rather than soda lakes, because soda lakes tend to be pH 9 to 10.
Yeah, I suppose. I mean,
Sorry.
Oh, go ahead, Sebastian.
That might not be a problem actually, because under the higher CO2 concentrations in the early Earth atmosphere that we talked about before.
As Eduardo said it maybe that would certainly affect that could affect the, as well as mineralization, then the pH of especially shallow bodies of water.
Yeah, there's an estimate in the Cohen and Catling 2020 paper for the pH of a solar lake under Hadean atmosphere based on the elsewhere modeled CO2 pressure in the atmosphere back then, which is somewhere between 6.5 and 9.
Yeah.
So that would fall in there. So it might not be exclusive. It might not be contradictory.
Okay, but as you say, they both mentioned the work by the John Sutherland Laboratory, and what's very interesting about that, the cyanosulfidic protometabolism, it explains essentially how all of the amino acids, or most of them, are made, and all as well as the pyrimidine nucleosides and nucleotides, and also the lipids. It also explains that glycerol is also formed. So glycerols are part of modern fat of modern sort of biological lipids. So it's a very, very interesting paper that series of papers that came out of John Sutherland's lab from around 2010 onwards. In particular, the 2015 paper, which it's in the which Seb, you've actually shared in the chat. Okay, but so it's nice to see all things coming from all sides. So it's good to see. This is really nice work, and it's you could have several conferences in the same year on this, and you'd have several parallel sessions running, and it would be but I think we are making great progress in coming up with a chemical explanation for the origins of life. I think in the last 20 years in particular, things have really moved on. We've moved away from the classical solution phase Miller-Urey experiments into involving minerals and rocks, which were ignored as basically inert inorganic accessories, and they were far from that. Hey, once again, thank you, Zack. Thank you, Seb, and David's gone, but he gave a very good introduction as well.
Thank you.
So I think that concludes all of our questions, and that'll conclude our session as well. So thank you, everybody, for attending. Thank you, Sebastian. Thank you, Zack, and David Catling left, but thank him too. And stay tuned for our next seminar. All right. Take care, everyone.